Thank you, Hugo. Good morning, everyone. Welcome from Seattle. I can see we have almost 2,000 people already logged in, with more counting up the digits every minute here. And I want to thank you all for joining us for this special section. On behalf of the co-organizers, we thought that it would be really important to include a session on this, in part because the reason that we are all spread across the world in our own living rooms instead of all gathered together in Washington, D.C., is this amazing small virus that has had so large of an impact. And despite all of the acknowledgement, of course, of the destruction that it's wrought, it has been interesting scientifically to watch how all of the advances that we have seen happen by coming to GSA meetings throughout the years play out in real time in addressing this viral threat. For example, just seeing the benefit of understanding evolution, of basic virology, of new advances in sequencing technology. It's all being deployed now in real time in order to combat this pandemic threat. It's really emphasized how global science is and continues to be, and how we continue to need the innovation from science in order to address our modern problems. In particular, our two speakers today are working on the front lines of addressing the viral threat. And for me personally, it's also been inspiring not just to see the amazing science that they're doing, but also the work they're doing in terms of public outreach to explain science to a world that wants to understand, but is also hit by lots of different sources of information, not all of whom are the most accurate. And so I want to thank both of them for taking time out of their busy schedules to join us today and tell us what the latest and greatest is that they're doing working on this virus. So first we have Richard Nair, and I will turn it over to him now. And just to describe the format of this session, we will have a talk by Richard and then a talk by Leah Starita. And then at the end, we will take discussion questions from the audience, which you can submit using the little Q&A button that's at the bottom of your screen. And then at the end, we'll take questions for both talks. Thank you. Take it over.
Yeah, thank you so much, Maitreya. Thank you to the GSA for organizing this special COVID session. I hope I can sort of live up to the promise by Maitreya. I certainly have some of the latest. I'm not quite sure I have all of the greatest. But sort of what I'm sort of trying to do here today is sort of a little different from the usual sort of scientific talk that I would give. Because much of what I'm going to speak about is not sort of work by myself or by my group, but instead I'd like to give you a bit of sort of more collective sort of bird's eye view of these efforts to track and trace the disease using sequences, from the perspective of NextStrain. That sort of seems more opportune for this purpose here. And for this reason, because it's such a collective effort, I would like to start out with the acknowledgements. So NextStrain, and that is really the core of this, this presentation, started as an idea between Trevor Bedford and myself in 2014. And we were thinking, wouldn't it be great if we had some sort of online resource where every week we'd have sort of a phylogenetic analysis of flu available for everybody to look at. And this sort of in 2014 was the seed of NextStrain. It has developed since into a platform where real-time phylogenetics is available to the community for a large number of different viruses. And over the years, there have been a large number of different people that were really instrumental for getting this off the ground. And this would be, first and foremost, James Hadfield, who has been sort of especially doing the front-end development of NextStrain for many years. And now in the response to COVID, it has been James, Emma Hotcroft, and Tom Sibley, among many others, who have sort of really sort of spearheaded this development, in particular, as Trevor and myself have been quite inundated with sort of other tasks and responsibilities. I'd also like to point out that the data that we analyze on NextStrain is not data that we generate. These are data that are contributed by scientists, clinicians, and institutions around the world. They are shared through a platform called GISAID, which is traditionally an influenza database that has sort of specific terms and conditions that facilitates data sharing with specific strings attached to ensure credit to the data producers, they also put in a huge effort in curating these data such that we have sort of high quality sequences to analyze. Now, before I go into the details, or well, into next strain and SARS-CoV-2, I'd like to just take one step back. For me, at least, it now seems that we've been in this COVID-19 SARS whirlwind for ages, and I can hardly remember what the days before. But if we look back at what happened, this is not that long ago. It is sort of three and a half months ago that there was an ominous news out of China that there were 27 cases of an unidentified viral pneumonia connected to a particular fish market. That fish market was closed just a few days after, and then things really started to pick up. So the virus was identified in early January, and the sequence, the entire viral genome sequence, was shared on January 9. And it was clear we are dealing with a relative of SARS. At the time, it wasn't clear yet whether this was actually transmitting between humans or whether this was just sort of a repeated spillover from animals. Right around the same time, we've seen the first cases outside of China, first in Thailand, Japan, and then sort of pretty quickly in Europe and the US. The US arrow is missing here, but the US also saw cases at some point in mid-January. So what are we dealing with? So this new SARS coronavirus is a beta coronavirus, and I'm showing you here a phylogenetic tree of different beta coronaviruses, which was produced by Trevor Bedford. And you have the new SARS coronavirus 2 down here in red, and it's sort of a relative of SARS coronavirus 1. But note the x-axis here is the scale. So the divergence here between all these different viruses is sort of 10, 20, 30, 40 or more percent. So this is a very diverse family of viruses, which not only contains the two SARS viruses, but also MERS, the Middle Eastern respiratory syndrome viruses, which were caused by repeated spillovers from camels, causing severe disease. But there's also two other sort of human coronaviruses in there that typically cause mild cold-like disease. And this is the human coronavirus OC43 and HKU1. These are all related viruses with rather similar genetic structure, but very different sort of presentation in terms of how severe they are and how they transmit. So now zooming into the SARS-like coronavirus clade, we see this picture here where the SARS-CoV-1 viruses are up here in yellow, and they are sort of about 30, 40, 50% identical to the SARS coronavirus 2 genomes, which are down here in red. The blue ones are sort of other coronaviruses that are sampled from various animals. And in particular down here, we see that the closest relative of the novel SARS coronavirus is sort of an isolate that comes from bat with the acronym RATG13. And that is about 96% identical to SARS coronavirus 2. And these are sort of various pangolin isolates that dominated the news lately. So what can we say just from this sort of broad brush analysis? So SARS coronavirus 2 is in the same family as these other beta coronaviruses, where some of them, these seasonal coronaviruses, cause mild disease, common cold, which sort of come periodically every winter and infect some fraction of the population. And the former, SARS coronavirus 1 and 2 and MERS, they cause a severe disease with a high fatality rate. SARS coronavirus 2 seems to spread much more easily than its relative coronavirus 1 or MERS, but is not quite as severe as these two. We currently operate with the assumption that there is an infection fatality rate of about 1%, which is about 10-fold worse than typical seasonal influenza viruses. This group of coronaviruses is found in many different animals, and we don't know at present what the proximal host is from which this virus has spilled over to humans. The closest one, as I said before, is isolated from bats. Just one note of caution, coronaviruses do tend to recombine quite a bit. So the trees that I'll be showing you always have to be interpreted with that in mind. Now off to next strain. So I'd like to just give a brief overview of the genome and then sort of walk you through the last sort of 2 months of what we've learned from genomes. So these coronaviruses, they have a fairly large genome. It's a positive strand, single-strand RNA virus with an almost 30,000 base pair genome, which is one of the largest known RNA virus genomes. The whole almost 20,000, more than 20,000 bases of this genome are devoted to sort of the replication machinery, and the structural and the accessory proteins only make up a small part of the genome. Now, in late January, when we sort of started analyzing this virus with next strain in a sort of more real-time fashion, the picture that we had was the following. So this is sort of a snapshot of next strain from January 26, where here on the left you see a phylogenetic tree with some 20-something isolates plotted on a scale where the x-axis is the number of mutations by which they differ from these viruses here on the very left, sort of basal viruses sampled in Wuhan, and the yellow ones here are from Bangkok. And what you see is that these genomes are essentially identical. There are a couple that are exactly identical, and there's a few that are just one mutation away from the base here, and then some 2, 3, 4, and the furthest one is 5 mutations away. There's a couple of clusters of identical genomes here. These tend to be sort of family transmissions or sort of otherwise clusters. There's some spread that is seen outside of China and these Bangkok genomes here. But basically, the first take home, these genomes are essentially identical and suggesting that they are emerged very recently from a single source. What you see on the left is the same tree sort of shown on a time axis, assuming an evolutionary rate typical for these coronaviruses. Much of this fine structure here is actually pretty uncertain. This has to be kept in mind. With so little genetic diversity, it's hard to actually pin down the exact structure of this tree. Many of these clades are very poorly or not supported. Nonetheless, it gives you a bit of an idea of how these viruses have been sampled through time and when their common ancestor might have been somewhere in November, early December. So the take home from these early lessons from sort of mid-late January was that it was pretty unambiguous that this outbreak originated from one source as opposed to being a repeated zoonosis from a diverse animal reservoir, that it was a rather recent emergence in November or early December. And as we'd expected, we see sort of family clusters showing up and the first clades to show up. Now, this then has picked up very quickly, and over January and especially March, we've seen an enormous deposition of data in databases, and often at really a sort of a very, very rapid pace. So what this graph produced by James Hadfield shows the sample date in turquoise and the database deposition date as a black dot. And often the database deposition is just a few days after the sample deposition, after the sampling time. And by now, this was sort of early April, by now we are at sort of 10,000 sequences. So it's really been sort of a flood of data coming in from all over the world, which has been sort of quite a challenge to stay on top of. So yeah, as I said on here, this is sort of the closest we've sort of come to our aim of real time so far. As of yesterday, so that's sort of my latest, the tree, it's already a subsample tree, sort of contains about 4,000, 5,000 sequences. And as you see here on the left, this is again the view with mutations on the x-axis. This is time on the x-axis. By now, most of these genomes differ from the basal Wuhan variants by about 5 to 10 mutations. There's some outliers. And you see here in the time-scaled view that these early, the color here is geographic region. The early genomes are mostly sampled in Asia, and the later ones here, as we know, in the US and Europe. So if you graph the genetic distance from the root on the y-axis against time on the x-axis, you see sort of a secular trend of these viruses becoming more and more divergent from the root, with a rate of about 25 mutations per year or 2 mutations per month. One has to also note, though, that the dispersion here is very high. So there is, these mutations often happen in particular clusters. They don't really come in very independently. So the temporal resolution that we get from this tree is actually lower than what just the rate suggests. One particular sort of triple mutation that I sort of wanted to point out here just as an example is the following, where we see on one branch, and this is this branch here leading to a particular European clade, we see 3 changes in consecutive position affecting 2 subsequent codons, which just highlights the fact that many of these mutations don't necessarily happen independently. Now, what does this tell us about how the virus spreads? So I decided that, this example from Iceland, so an Icelandic group from Decode, they sequenced a lot of SARS coronavirus cases in Iceland, and one particular, and they make a nice example of how this virus has gotten around. What you see here, all these sort of dots correspond to genomes sampled in Iceland, where everything else is hidden in this graph. And the first thing that you see is that Iceland essentially uniformly sampled the planet. So every major clade is represented in Iceland. And individual clades, like this one down here, I've zoomed in here on the right, there's 5 sequences from Iceland, which are sort of nested in a cluster from, which is otherwise dominated by the US, other sort of American countries, and Oceania. So this then happens to be an introduction into Iceland somewhere from the Western Hemisphere. And by a similar token, you can sort of identify these clusters in local communities. You can identify clusters of spread through genomics. So just another sort of intermediate summary. SARS coronavirus 2 was disseminated widely before travel restrictions were put in place, and these border closures had very little effect. They're mostly sort of political. The virus typically spread locally before it was noticed, and this happened sort of during a time when testing strategies tended to be still focused on people with particular exposure history, particular travel history to known hotspots. So, and by sort of maintaining this focus for a long time, this local spread was often missed. The temporal resolution that these genomes give us is about 4 weeks, which is too low to resolve direct transmissions or directionality of transmissions, but it is sufficient to connect different outbreaks and to identify clusters, which can help epidemiological investigations substantially. We currently don't have any solid evidence that these mutations have functional significance, but if you are interested in sort of more in-depth analysis of this, I suggest you go to Sergei Kovakovsky-Pon's analysis of signals of positive selection. So before I end, and sort of Maitreya already sort of pointed this out in her opening statement, this has also been an interesting struggle and experience in science communication and in sort of keeping the lid on misinformation. And this has had sort of multiple aspects to it. And one of which was that there's all sorts of conspiracy theories and sensationalist research that has been sort of published or pushed in the media. And I've just put up a collection here, which have been sort of the most outrageous ones. So there's one which keeps coming back, and that is COVID-19 has been with us for much longer. It's not so bad after all. Why all the fuss? This is manifestly wrong. The genetic diversity very clearly shows that this is a recent emergence, and respiratory samples from last year are uniformly PCR negative for SARS coronavirus 2. There were some sort of more hilarious ones, like that the origin of this is snakes, which was based on some misguided sort of codon usage analysis, was sort of debunked by Christian Andersen. There was one very widespread one that it might be some recombinant with HIV. This happened to be a paper that had for at least for some time, the highest altmetric research attention score ever. This was solidly debunked by all sorts of people, including Trevor Bedford. Then there's all other ones that are likely wrong and keep surfacing, often sort of pushed by authors that are looking for lots of attention in the media, often also given some undue authority by the fanciness of their affiliations. So there's one of the prevalent serological studies, the seroprevalence studies that we've seen in recent days. They claim prevalence is high when the evidence for this is actually very shaky and often produced by sort of not correctly accounting for high false positive rates in their tests. Then there is repeated claims that these viruses are, there's different variants, some bad, some not so bad. And these are usually easily explainable by sort of various confounders in the data. So at the very end of my talk, I'd like you also just to look a bit into the role Nextstrain has played both in the positive and not so positive ways in generating these kind of misinterpretations and misinformations. And what we've had to learn sometimes in a rather sort of painful way is that the colorful, beautiful trees that we share are also very susceptible to misinterpretation. And this has certainly been a very steep and rough learning curve for us. And I've shown you one example here where I've zoomed into a clade on Nextstrain where that is dominated by European sequences and has 2 Italian sequences sampled in Italy early on in January, which sit at the base of this clade. And then there's a whole bunch of genomes which are descendant in this clade that are also sampled in Europe, and many of which sort of trace back to Italy. So, you might jump to the conclusion that Italy, actually, that these early genomes seeded this entire outbreak. However, this is most likely not the case, even though this seems like very compelling and very plausible, because we know, for example, that the various genomes in this clade, like this one here, actually have a documented exposure history in Asia. So it is this clade was introduced probably multiple times into Europe, and it is impossible at this stage to look at a tree like this and sort of draw as a direct directionality, a direct sort of connection between 2 such things. So what one has to keep in mind, and what we've had to learn sort of how rapidly such misinterpretations can spread, is that the low genetic diversity in these data combined with very biased sampling means that you can't sort of get at any directionality or sort of specific statements beyond that these different outbreaks are genetically connected. So we've tried to sort of prevent this sort of to happen by sort of creating Nextstrain situation reports, sort of basically by capturing the narrative and giving a guided interpretation to what you can read off the phylogenetic trees. This is currently slightly problematic because GitHub seems to be down, which is serving this. But I'd like to, if you have a chance, have a look at the Nextstrain situation reports and see how you can use this interactive display to walk through what's going on. This effort has been led by Sydney Bell and Cassia Wagner, together with Emma Hodcroft, James Hadfield, and Nicola Müller, and many others. Okay, this brings me to the end, sort of my final acknowledgement slide. Again, yes, this has been a fantastic collaborative effort with Trevor Bedford in his lab for many, many years now. I'd also like to thank all the people at the front lines, all the healthcare staffs that take care of these of the patients that come down with this terrible illness, and everybody else who's sort of keeping the shop running. Everybody else who needs to work, needs to go out and expose themselves to COVID-19. It's really been a humbling experience. Yes, so the data, I talked about that. Thank you all for your attention, and I'm sort of happy to, I guess, questions we do after the second keynote. Thank you very much.
Great, amazing work. Thank you for that. Next, we will have second presentation in the session from Leah Starita. So Leah, you can go ahead and share your screen. And I've noticed some people raising their hands in the interface here, and unfortunately that doesn't work. So what you want to do is hit the Q&A button instead if you're asking a question. Great, Leah, take it away.
Okay. Is this working?
Yep, looks great.
Okay, so I want to start with some receipts. So I was asked to give this talk about 2 weeks ago. And I said, can I use expletives? Because I'm pretty sure that this story can't be told without them. And Mark Johnston didn't think you all would care. So here we go. Hold on. Okay. So I've gotten many emails over this week since the talk was announced, asking what the heck am I up to, right? Don't you work on human genetics? How did you get involved with COVID testing? And so I told these folks that I had a side hustle where I was actually doing the molecular work for a project called the Seattle Flu Study. And all of my part in this started when my friend and colleague, Trevor Bedford, who you just heard about from Richard Nair's talk, said, hey, Leah, do you think you could sequence a few thousand flu genomes? You know, and this is kind of how a lot of people know Trevor Bedford these days, as, you know, his other 200,000 Twitter followers. And I was like, well, do you mean like load a NextSeq? I can do that. I can do that in my sleep. Sure. And so this is where it all started for me anyway. But it actually had started a few years before that. So this is Bill Gates. He famously gave a TED Talk in 2015 about how, you know, the world was kind of woefully unprepared for a pandemic like we're seeing right now. So a respiratory disease that is, you know, spreading from person to person, and how the world was really just not prepared to detect it or to really do anything about it. And as we know, he was right, but Bill wanted to do something about it. So he galvanized, or funded, the Seattle Flu Study. The study was organized and launched in the fall of 2018 with a mission of transforming how infectious disease outbreaks are detected, monitored, and controlled. Since there was not a pandemic in 2018, the idea was to use the annual emergence and spread of seasonal flu as a model. So we were to build the infrastructure needed to heavily sample acute respiratory infection cases in a major metropolitan area, and we were all in Seattle, so that's the city we picked. We had lofty goals to learn to monitor a pandemic using advanced pathogen detection and sequencing, build actionable flu maps and models, do genetic epidemiology, as Richard Nair was just kind enough to tell you about through NextStrain. And so this vision required a very big team and lots of innovations across the board, especially in recruitment to get that many samples, high throughput testing and sequencing, the data warehouse that Trevor built that this all runs on, and many more. But first, I'm going to talk to you about how we actually got these samples, right? So we're supposed to deeply sample respiratory disease in Seattle. And so if we're going to do this, we've got to get the samples. And so we have three clinicians who have been innovating in this area. So first of all, the first way we got, so this was led by Helen Chu, who really spearheaded a lot of this for collecting samples from adults. And then Jan England from Seattle Children's Hospital, who knows anything and everything there is about respiratory disease in children. And Michael Book from the Fred Hutch, who studies respiratory disease in immunocompromised people. And so the first way we got samples was the easiest, and you would think wouldn't take a lot to negotiate, but it did, is medical dumpster diving, really. So taking the remnant or leftover samples of respiratory tests from hospitals, right? So a lot of times the hospital will only test for flu and RSV, but we would, look for more pathogens in those tests, in those leftover samples. So we did this for the Seattle Children's Hospital System, which is pretty massive, and then also UW Hospital System and other area hospitals. But we also wanted to be able to capture people who weren't seeking medical care. So we had pop-up kiosks in heavily trafficked areas, such as the Student Center at UW. And we really wanted travelers, right? So as we've seen over the last couple of months, travelers can be really important to capture. And so we were in light rail stations, we were at the train station, we were at the airport, and other areas where tourists would hang out too. So we were down at the Westlake Mall and things like that in downtown Seattle. But the recruitment avenue that would become the most important later is the Swab and Send program. And here on the right, this is an article written about it. So we had a lot of targeted advertisements for this so that people would be aware of it, right? So how your family or how you can help fight the flu from your couch, right? So the whole point of the Swab and Send program was that people would never have to leave their house, right? They wouldn't have to leave their house, spread whatever they had all over the place, and we could find out kind of what they had without them, having to seek medical care. So if somebody was sick, they could go to seattleflu.org. And again, we had a lot of targeted advertisements through Twitter and Nextdoor and Facebook, so that there was an awareness of this program. We would send them a testing kit by delivery service, and usually it would get there in about 2 hours. And you can see this screen grab of this fella here. He's got his swab, which is right here. I don't know if you guys can see my mouse. And he's got the transport media, and he's got his little biohazard bag. So what our participants did was they would get this kit, swab their own nose, and then package the sample back up. Package the sample back up and send it back to my lab at the Brotman Beatty Institute at the University of Washington. And we, as I said, we did a lot of advertising for this on social media to raise awareness, and we wound up collecting more than 3,000 samples this way over the last year. So all these sample streams send their samples back to my lab for processing, but we needed more firepower in terms of automation and integrated LIMS system. So we called on Debbie Nickerson of the Northwest Genomic Center at University of Washington to help us deal with a project of this size. And we would end up sampling 23,000 samples just in the first 10 months or so. The first step in pathogen detection, and this will become important again later, is RNA extraction, right? So we have an automated extractor. This is a Roche MagnaPure 96. This thing is a beast. It can crank out 96 extractions every hour in 10 minutes. It's pretty awesome. And then we have to find out which pathogens are in the samples. And so and this is Debbie's idea, what we did was we employed Thermo's open array TACMAN system. So with the open array, the TACMAN assays are actually arrayed on this chip. This chip, there's 48 samples. Each one, there's 56 through holes for each sample. So we could actually test for 27 different pathogens. Some of them are multiplexed. In duplicate, in each one of these little sets of 56. So we could do 48 samples on this chip alone. The machine can hold 4 chips at a time, so we can run 188 samples at a time. So about every 2.5 hours, we can run another 188 samples for and query the presence of 27 different pathogens. And these pathogens are, all of we called it the flu study, but again, everybody thinks they have the flu when they have an illness in the winter. So the panel contained influenza A, both subtypes H1N1 and H3N2, influenza B, parainfluenzas, RSV A and B, rhinovirus, and then it also had the seasonal coronaviruses on it, enteroviruses, and some bacteria as well. And of course, a human control, right? We always want to make sure that the person actually stuck this up their nose. Okay, so using this panel, we could see basically what everybody had all winter, right? So what we're looking at is just the prevalence of, or the case count of different respiratory viruses over about a 6-month period from last October to March 1st. Trevor actually used this to debunk some theories that, everybody had coronavirus back in December, and we're like, no, you had flu B. So you can see in light blue, there's flu B, and you can see it rise early in the flu season last year. We had a weird flu season. It really peaked in the before the holidays, and influenza B actually usually follows influenza A, but it came first last year. And you can see a lot of people have influenza B, they have rhinoviruses, and RSV. Okay. Another person who was really important for all of this work is a guy named Mike Famulari at the Institute for Disease Modeling out in Bellevue. And one of the things we really wanted to be able to build using all this kind of data was an accurate map of where the flu was in Seattle. And we were hoping that this could become actionable, right? So maybe the hospital systems could use it, or school systems even, to see where flu is spreading and take steps to mitigate it, right? And so here, this looks like it's what was happening in Seattle on January 3rd of 2019, and you can see that flu is kind of concentrated in the islands and in Western Seattle, and gets less prevalent as you go east toward the mountains. This is an island kind of famous for not vaccinating, so it's really not surprising. Okay. We were heavily sampling influenza and other respiratory disease in Seattle in 2019, when we had a historic snow event. Now, it very rarely snows in Seattle, so when it does, it completely cripples the city, and we got something like 2 feet of snow over 10 days. Kids were out of school for 2 weeks. Back then, we thought that was really terrible. People stopped commuting to the city. I think traffic was down by 60%. On I-5. And what you can see in these gray lines, so Mike is modeling what should have happened in the gray lines here. You can see flu A come up, flu A, H1 go up and come back down. Same with H3, RSV, seasonal coronavirus. But with the colored colored lines, you can show what actually happened. And so you can see that flu A H1 was basically reduced. The H1N1 epidemic in the city actually completely stopped after the snowpocalypse. And then the H3N2, which should have happened earlier and been a little worse, was actually attenuated. And the same for RSV A and B as well. And so what we're seeing here basically is that, social distancing forced by a major snow event is useful for slowing the spread and extent of respiratory disease. And, again, this comes in handy a little bit later. Okay. So, but we didn't only detect influenza, we sequenced it too. And now I am glad that Richard just spoke before I did. So you guys all now know that I am showing a screen grab from NextStrain with flu genomes for flu A, H3N2 on the left, and flu B, Victoria on the right. So the red ones are flu genomes sequenced in Seattle. Through the Seattle Flu Study, we were actually producing 15% of the world's flu genomes. Unfortunately, this data is not in the public version of NextStrain, but we hope Trevor finds the time to release it eventually when he's not trying to debunk tinfoil hat conspiracy theorists. From the dates on the bottom, you can see that there was a lot of H3N2 in the winter of 2018 and 2019, and then barely any in the 2019-20 winter. The opposite is true for flu B, where you can see that there was a lot of it in the winter of, or not very much at all in the winter of 2018-19, but it predominated in the flu season of 2019-20. What you can also see is that there's many introductions into Seattle over the course of the season, and that what happened up here in H3N2 in the very top, where you can see these huge transmission chains of H3N2, was that this was a vaccine mismatch. So these H3N2 strains that landed in Seattle were the ones that were able to spread more easily. And then what you can see in the flu B here on the right is that there are many introductions of flu B, and many of them were actually able to expand into large transmission chains. And, you know, this kind of looks a lot like the data Richard just showed you, where there's many different sparks, and then only some of those viruses are able to cause to transmit in the population. Okay. So you'll notice that these all stop at about January 2020. And you can imagine what we all started paying attention to, right? So I remember being with Trevor at dinner in mid-early January, and he says, you know, this virus in China is definitely spreading human to human. We have a few weeks before it gets here, and it seems to be really bad. Sometimes going to dinner with Trevor can be a real bummer. But anyway, as I just told you, we had built this huge respiratory surveillance platform, and of course, we're going to use it, right? So now we get into detection and sequencing for SARS-CoV-2. So now is the moment all of us molecular biologists have been training for our entire careers, right? We're going to design some primers, we're going to do some PCR, and we're going to find some virus, because that's what we do. And quite honestly, it's not that hard. But first, we have some decisions to make. It would be great if we could have just added a SARS-CoV-2 assay to our open array design, but these things take 4 to 6 weeks to manufacture, so that's out. Next, the CDC had just published their assay, but who in their right mind would design an assay with the throughput of 20-odd samples at a time? There's pretty clearly a pandemic coming our way, and they didn't multiplex any of these probes. They required a special thermal cycler. Like, what the heck were they thinking? And yeah, 22 samples at a time they can do with the controls in their plates. That's insanity. Anyway, so we wanted to keep running the open array pathogen panel. So we need to be able to run at 190 samples at a time. So we're not going to do the CDC assay. So get this, I didn't even have to design the primers myself. I called the experts at Thermo, who design TaqMan assays for a living, and they sent me a probe set with a limit of detection about 6 molecules per reaction, no cross-reactivity with coronavirus, And so we multiplex that with an RNase-P target. Again, we need a target that tells us that there's human RNA in there too. We multiplexed it with a different fluorophore so that we could run the assays in the same tube. And we can also run them, so we did 190 samples in duplicate at the same time in 384 well plates. So we've got an assay and we're ready to go. Okay, so we start running all of these respiratory samples that our clinical team has developed all these cool mechanisms to collect. I'm not going to tell you when, but let's just say it was later than we would have liked. So when we started, you know, we'd only tested for a couple of days, and I ran into Danielle, one of our technicians in the hallway in genome sciences, and she's looking at me kind of sideways, and I'm like, uh-oh, what's going on, Danielle? And she's like, we got one, right? So again, you know, this is before there was really a heck of a lot of testing going on in the United States. And, you know, there's very strict rules for the CDC. You have to have traveled to China or known somebody who traveled to China to even get a test. And so, you know, she says, well, there's one, and I go and look at the machine with her, and clearly these bands, I mean, this was definitely a positive. We confirmed it using the CDC primer sets, sent a sample to the state lab, and it turns out that we had just identified the first first documented community-acquired case of COVID-19 in the United States. It was a teenager from north of Seattle. Turns out he was feeling better by the time the state confirmed the case, but public health officials were actually able to pull him out of high school just as he was heading back in after being out sick. So we kind of call that one a win.
Yeah, sorry to hurry you up a little bit, but we're almost running out of time, and I want to leave some time for questions. So I'll give you a couple more minutes, but we should try to.
Okay. So as I said, this is Washington 2, kind of riding shotgun here to the state lab. All right. So using NextStrain, Trevor was able. So we clearly sequenced this virus as fast as we could, and using NextStrain, Trevor notices that this virus is clearly very likely descended from the very first case of coronavirus found in the United States from January 19th, right? So this is now February 29th. This thing has been spreading in Washington undetected for 6 weeks. So it looks like there's kind of a big epidemic on our hands. So the Seattle flu study then goes through a metamorphosis through the swamp of regulatory morass. And then we came out the other side as the Seattle Coronavirus Assessment Network, where in collaboration with Public Health King County, we've repurposed our Swab and Send program to do community testing of the population. Okay, so more important for this crowd, and I would like to get you guys interested in this, is how to ramp up testing, right? So we've got major problems. If we're ever going to get out of our house, we have to be doing millions of tests a day. So how are we going to do that? We've got massive supply chain problems. So first, you need swabs and transport media. Can't get any of those. RNA extraction reagents. It's really hard to come by those. And then there's RT-PCR reagents. Thermo happens to like me and hasn't throttled my TACPath usage yet, but I have a feeling it's coming. And I'm constantly, nearly running out of things, even like primers. It seems like the companies are having a really hard time keeping up with that. So we're trying to figure out ways to kind of cut pieces out of this pipeline, right? So we were figuring, well, maybe we could use dry swabs, which would cut out the UTM, right? We just paid 8 times what we normally pay for UTM from BWR. Thank you, BWR. And what if we could then cut out the extraction? Maybe we don't actually have to purify the RNA. We would cut 2 of the major problems of the supply chain out of our pathway. So we did those experiments, and through the study, through a study run by Helen Chu, we're actually able to find COVID positive patients that have just come out of the UW system and chase them down and kind of ask them for another swab. And so we did that, and we showed, this is Sanjay Srivatsan, and he's a graduate student of Jay Shindure's, and what he showed was that taking these dry swabs from people, so they didn't actually arrive in the lab in the typical media, and just extracting it in plain old, good old TE, had exactly the same sensitivity as doing like the classical RNA extraction and going, so we did the dry swab, eluted it in TE, and then right into RT-PCR, and it works just as well as actually doing the extraction. So this paper is actually come up on bioRxiv today, and there's a lot more work to do, but we wanted to get those preliminary results out there. Sri Kozuri has built a COVID testing scale-up Slack. You can check out his Twitter account for the link. It's public for any scientist to come and join the conversation. And there's a lot of great ideas bouncing around on there, and some working groups that have formed. And it's a really neat science, genomics coming to the rescue story. So I encourage you all to check out that. And of course, there's a massive team. So just the PIs on the left from University of Washington, Fred Hutch, and Seattle Children's. And then on the right, this is now just my team here, and part of Debbie's, who is running the testing now for the Seattle Coronavirus Assessment Network. Thank you all for listening.
Fantastic, great, thanks so much. Richard, if you could go ahead and come back on camera. As you guys might imagine, we have a bunch of questions, and so we're certainly not going to get through all of them in the next 10 minutes, but I want to let everyone know that there's a new Slack channel called COVID, where you can go in and talk more about these results and others and talk to each other. And so Richard, the first question I have for you is summarizing a bunch of different questions that we've gotten over the chat, and it's about the mutation rate. I think as geneticists, we always like to see mutations that do things, right? And so all these mutations that don't really have a functional consequence are interesting, but hard to think about for some of us. So how long do you think it might take before some of these mutations start having functional significance? And do the mutations that are more drifty already have some kind of a consequence for things like vaccine development and transmissibility and things like that?
Yes, so we don't actually know, right? There's certainly several mutations that have been that happened independently in different parts of the tree. So there is some reason to believe that they are, that it's either sort of a very high mutation rate locus. There are some places in this genome which seem to be very slippery, very low complexity regions. Some of them might be selected for something or another. It is just there's many publications or many claims that jump onto these and then make a story out of them, which is often very poorly substantiated. So, all I'm saying is that the evidence for them having particular functional consequences is very weak. I think evidence that some of these are happening in a more systematic, positively selected, potentially way is better. But further research will show. So also just dumping them on a virus cell culture is not going to do the trick, because replication in a cell culture system is very poorly generalizable to how this onto the pathogenicity of this beast. So one probably needs better systems, better models to study the effect of these mutations. The jury is just still out. And people are still trying to, make headlines with this, and I think that is dangerous.
Speaking of kind of dangerous questions, I thought I'd ask you your advice on how to talk to people about this theory that the virus originated in a lab, because that does seem to be one of the more common conspiracy theories out there. And in fact, I just told a repairman yesterday who came by my house that that's probably not what's going on, but he seems still skeptical. So do you have some talking points that we as emissaries of science could use to talk about those theories?
Yeah, so I left it off that slide. So, it is not a terribly plausible story, right? So we know there's lots and lots of coronavirus out there, and we have isolates from bats, which are very, very similar to this guy. And the chance that via some intermediate host, this is sort of an animal-human transmission, it is sort of the more likely, so the more plausible explanation at this point in time. So we don't have any serious evidence to the contrary. So it's something that people like to think, and it's sort of a very juicy story, but the evidence for it is simply not there.
A question for Leah. This is truly amazing, the scale that you guys are operating at. And again, multiple people have asked questions along these lines. What are you doing to try to get other labs to work in the same way, to spread the scan model across the world?
Well, actually, we've shared our systems with everybody. Anybody who asks gets copies of our IRB, they get pictures of the kit, they can have anything they want. So that's, first of all, is just open source sharing. We've shared our testing protocols, everything. It's all online. If you go to my Twitter account, basically you can click on our protocol. Everything is there. We are encouraging, there's actually a program that's going to spin up at UCLA using almost a clone of our kit right now, which is pretty cool. I think right now, though, we really need to think about scale, right? So the way that we're doing things right now, the big companies like LabCorp and Quest can do this, but, genomics labs like us, I employ 10 people just to rip boxes apart. That's not sustainable. So what we need to be doing is thinking about how to do this smarter. And so if we're thinking about, can we hook this up to sequencing on the back end, things like that, so that we can be multiplexing, tens of thousands of samples and not just a few hundred.
Fantastic.
So we're working
what
on
are
that.
your thoughts on the prospects for the antibody testing for serology? That seems harder.
It is harder. I don't really know a heck of a lot about it, so I'm going to leave that off. But I think that people should think about Bayes' theorem when reading about serology tests. And I'm just going to leave it there.
Great. You said something that was interesting, which is that you have to do a human control. Is that because partially it's difficult to do the swab yourself? I've heard that it's pretty unpleasant. And also, are people trying to swab their pets or anything like that? What are the main compliance issues that you're finding?
Oh my God, the compliance issues. You have no idea. These people surprise us every day with their inability to put their name on a tube. And we have to throw them out, which is really sad when swabs are like gold. Anyway, yeah, so we do the RNase-P, so it's just a highly abundant human protein or human gene. And we do that just to make sure that there's enough material on the swab. So we say we have a cycle cutoff in the RT-PCR to say, okay, well, this is kind of a good enough swab, and we'll count it as a positive test, just to make sure they actually stuck it up their nose. We have people that there's no RNase-P on these samples, and I'm like, well, did you chicken out? What? So it is actually a really important control to make sure that it's not a false negative.
Great. I'm not sure which one of you might be better equipped to answer this question, but we've gotten several questions about the human genetics side of things. So there are certainly a lot of human geneticists trying to do association studies or other tests to figure out if there are human genetic differences that lead to susceptibility differences. What are your thoughts on the current state of that? And are there people we should be following their work that are doing particularly nice work along these lines?
I think I can take some of that. So I know the UK Biobank is definitely working on this. And there's a consortium here in the US as well. For us, the part of the biggest problem is getting genetic consent. We've always been well, I work with Debbie Nickerson. She's just think, we've got human genomes here. What are you doing? And, just trying to convince our clinicians to try to get human genetic consent, right? That adds a whole new layer to our consenting process. And that's been kind of really difficult to convince people to do. But I think there's going to be a lot of studies on, how people have reacted and the severity of the disease, given their human genome, I think is going to be really interesting going forward. And I think Richard has something.
Yeah, no, I fully agree, and the UK Biobank is probably best equipped to get there. These studies are complicated by the fact that we have these massive dependence on age, right? And together with founder effects in different communities, it is very difficult to disentangle sort of confounders here from signal. Also how sort of healthcare seeking behavior changes dramatically. So there's a lot of very, very difficult confounders which get exponentially amplified in the process of these outbreaks, which needs a lot of data and very careful data to control for this.
Great, thank you both very much. I'm told the system is automatically going to kick us off at 8:20. So I think we will end it here. And I want to encourage everyone to continue answering and asking questions in the COVID Slack channel. Thank you both so much for the work you're doing and for taking time to talk with us about the latest and greatest. This was a fantastic session. And thank you all for joining us this morning.
Yeah, thank you, Maitreya.